audio recording of lecture 6, positive RNA virus. The replication of positive strand or sense strand RNA viruses. Slide number 2 is a depiction of various classes of viruses based on Baltimore classification. And their modes of replication. Class 1 double-stranded DNA viruses, class 2 single-stranded DNA virus, class 3 double-stranded RNA viruses, class number 4 positive RNA viruses, class number 5 negative RNA viruses, and class number 6 those RT viruses. And we will discuss different examples of different classes of viruses throughout the lectures. So slide number 3 talks about RNA virus replication, those viruses that their genome is RNA. So basically, the way it works is that we have a process called replicative intermediate, or RI. We have two steps of it, replicative intermediate 1 and 2. And what happens in all of these RNA viruses is pretty much the same, follows the same format, where whatever your genome is serves as a template, and this template makes an anti-genome or an opposite sense. For example, if we have a positive sense RNA virus, it makes a negative sense RNA virus. If we have a negative sense, it makes a positive sense. So by the end of replicative intermediate 1, we have accomplished the production of the opposite sense of the template. And during RI2, or replicative intermediate 2, whatever anti-genome that has been made during the first stage will serve as a template, and we make new genomes. So if we have a positive strand RNA, during the first step, we make a negative RNA. During the second step, we make more positive. So we end up with an accumulation of both positive sense and negative sense RNAs. Slide number 4 is basically a more detailed description of this process of replication of RNA genome viruses, that I would recommend you guys to take a look at it and make sure you go through it. Slide number 5, a family of positive sense RNA viruses is called Picornaviridae. Pico, small, RNA is just basically the genome. So these are positive RNA viruses, non-enveloped, and they have an icosahedral capsid. As we can see, we have the capsid proteins VP1, VP2, VP3 in the cleft, that they also serve as a viral ligand. Slide number 6. A genus within the family of Picornaviridae is enterovirus, intestine. So enteroviruses is a large group of related viruses. These include poliovirus, echovirus, Coxsackie virus, and enterovirus. And for poliovirus causes poliomyelitis. Echovirus causes enteric cytopathic human orphan virus. And in general, the genus enterovirus, they cause upper respiratory tract, gastrointestinal tract, central nervous system, heart, muscle, skin, and they are stable as the pH changes. So pH changes cannot cause any damage to the virus. A second genus within the Picornaviridae family is called rhinovirus, rhino nose. So we have over 100 serotypes of human virus, 100 serotypes of human rhinovirus. And these are mainly those viruses that cause common cold. So there are common cold viruses. They're being transmitted via aerosols, direct contact of infectious secretions, such as hand to mouth, nose, eyes. Those secretions are capable of transmitting the disease. The virus replicates in oropharyngeal mucosal cells. They are acid pH labile, so they are sensitive to acidic pH. Therefore, they cannot be found in the GI tract. And adults, on average, get to 3 to 4 different infections of rhinoviruses per year. Another genus is called Aphthovirus, ophtho ulcer. It causes a disease in livestock called foot and mouth disease. So a very prime example of aphthovirus is foot and mouth disease virus, or FMDV, that can infect cattle, sheep, pigs, and goats. Slide number 9, another genus of Picornaviridae family of viruses called cardiovirus, cardio heart, and it infects human brain, muscle, and heart. And in mice, it causes encephalomyocarditis. So there is a virus, a mouse virus within this family called encephalomyocarditis virus, or EMC. That's the name of the disease, encephalomyocarditis. Slide number 10. Another genus of Picornaviridae is the genus Heparnavirus, hepa, liver, RNA viruses. A prime example of a virus within this family is called hepatitis A virus. It is an infectious, causes infectious hepatitis, and the hepatitis A virus is very difficult to grow in cell culture. Transmission of hepatitis A virus is via contaminated food or water. So let's slide number 11. Let's start by taking a look at the genome of this positive RNA viruses. Example would be poliovirus. As we went over, the poliovirus capsid is made of these capsid proteins, then they form pentamers, etc., etc. But now we are talking about the RNA of the virus, or the genome of the virus. The genome of the virus is 7.7 kilobase. It has a single open translational reading frame. it has one single open reading frame at the center of the genome. The 5 prime end of this positive RNA does not have a uh, 7-methylguanine cap, but instead it has a small viral protein, VPG, viral protein G. That this region serves as a primer for the transcription of the genome. And at the 3 prime end of the genome, we have a poly A tail. Remember that positive RNA genomes always serve as messenger RNA. So any virus that has a positive RNA genome is called infectious RNA. Let's talk a little bit more about RNA genome expression, slide number 12. As we discussed before, poliovirus gains entry through receptor-mediated endocytosis. There would be uncoding, the separation of virus, capsid, and genome, and the release of RNA or the genome of the virus occurs in the cytoplasm. As we just mentioned in the previous slide, poliovirus genome does not contain a 5' cap. However, RNA attaches to host cell ribosome by a recognition sequence called IRES, or internal ribosome entry site. Remember, I mentioned in the previous slide that positive RNA genome can serve as messenger RNA, so it can basically be translated. In order for translation to happen, the genome needs to attach to a host cell ribosome. This is usually directed by the 7-methylguanine cap at the 5' end of the positive strand RNA or the messenger RNA. Since this genome lacks that 5' cap, the recognition of host cell ribosome is accomplished by a sequence within the RNA called recognition sequence, which is also termed as internal ribosome entry site, or IRES. And after binding to the ribosome, viral messenger RNA starts being translated to make one large polyprotein. Slide number 13 is a little bit more detailed and technical description of the poliovirus, which is a typical picornavirus. Slide number 14, for those of you who may have forgotten about IRIS from previous courses, I guess we discussed and covered that during microbial genetics. But anyways, internal ribosome entry sites are those sequences within the RNA that can recruit the translation machinery of the host cell. This translation machinery is the ribosome. And this process of recruitment is independent of the 5' end of the messenger RNA. Slide number 15 talks about poliovirus. Polyprotein, multiple proteins. So as we just mentioned, that this because of the presence of one single large, about 7 kilobase open reading frame, and because of the nature of the RNA, that is positive RNA can serve as messenger RNA, it makes one large polypeptide chain, or one large protein. One single protein will be cleaved, will be chopped off into three precursor proteins. The process of chopping off is accomplished by autocatalytic action of viral proteases. So it can happen by itself. It does not require any extra stimuli or signal or any other machinery. So this single protein is cleaved by autocatalytic viral proteases. The two viral proteases are 2A and 3C, and they cleave at specific regions. Once they cleave it, we will end up with three precursor proteins. One P1, another one is P2, another one is P3 from N to C terminal region. These three P1, P2, P3 segments will be further cleaved by viral proteases to make tons of additional proteins. For example, P1 can make VP0, VP3, VP1. P2 can be cleaved to make 2A and 2B and C. 2A is a protease, so it participates in a positive feedback loop. Then P3 will be cleaved to make 3A, B, and 3C and 3D. Remember, 3C is again another protease. So this process will continue itself. So, slide number 17 talks about poliovirus transcription of complementary RNA. Remember, we said that it happens in two phases: replicative intermediate 1 or RI1 and RI2. During RI1, we have a genome that is positive RNA. This genomic positive RNA will be transcribed to anti-genome or negative RNA strand. We need to make a complementary strand. So now we have a positive strand and a negative strand. The transcription complex that facilitates this process is basically we have at the 3' of the positive RNA genome, we have poly A tail, we have the viral protein G located at the 5' end of the genome that serves as primer for making negative strand. Also, some host proteins are required for this transcription to happen. Those are called transcription factors or TF. These are proteins. We also need an enzyme within the virus called viral RNA replicase. That are it's basically a viral protein 3D that in the previous slide we discussed that it will be generated by alkylated cleavage and further processing of the single protein. So this viral replicase helps new genomic RNA synthesis in a 5' to 3' direction. So that's basically RI1. What about RI2? During RI1, we made a lot of negative RNA or anti-genome because the genome was positive RNA, we made negative RNA, and this negative RNA produced during the first phase will serve as a template to make the complementary strand during the second phase. And this complementary strand is what? Is a positive sense RNA. Again, we need to have the transcription complex. We need to have some sequences at the and the 3' end of the anti-genome. We need to have the viral protein G. We need to have host transcription factors or proteins, and we need to have viral RNA replicase. This newly synthesized positive strand RNA genome serves two things. Number one, I initially mentioned that these positive RNAs can serve as messenger RNA. So this can be translated into one single protein, and then that will be cleaved. So we make more viral proteins, both structural and those enzymes required for viral replication. And this positive RNA that is made during the second phase can serve as the genome for new virions. Remember, the viral genome is positive strand, so we need to have more positive RNA genome. Why? Because we need to use some of these molecules as messenger RNA and some of them as the genome for new virions that are being produced. Slide number 19 is a depiction of the poliovirus RNA synthesis. Each virally infected cell, after the virus gets in through receptor mediated endocytosis and it being penetrated and uncoded and starts replicating, it makes about 4 times 10 to the power 5 RNA molecules. So about 400,000 molecules. And off of these, about 10% are negative sense and 90% are positive sense RNA for the two reasons that I mentioned in the previous slide. We have discussed slide number 20 a couple of times before. Poliovirus assembly, the viral proteins VP0, VP1, VP3 are being produced as a result of autocatalytic cleavage of the single protein and further cleavage. So we had these three called protomers. They come together. 5 of these complexes of 3, 5 times 3 would be 15, will form pentamer. And pentamer, then 12 pentamers will form a procapsid. Procapsid is when the RNA will be encapsulated. Then we have 12 of these 560, 60 protomers that are so-called, they look like a headful insertion of viral RNA genome during this stage. And after that, we have a virion where VP0 is being cleaved to VP2 and VP4. So a complete virion has VP2, VP4, VP1, and VP3 as its capsid proteins and positive sense RNA being encapsulated in the center of this poliovirus. So poliovirus after infection causes host cell cytopathic effects or CPE. And what are these? Very soon after viral infection, host cell translation of the proteins will be stopped, usually about 30 minutes, half an hour after poliovirus is being has been successfully infected the host cell. Then viral proteases, those proteolytic enzymes that come in with the virus, activate host cell proteases. And these host cell proteases will cleave a 222 kilodalton ribosome protein called eukaryotic initiation factor 4F. So virus comes in, 30 minutes after infection, host cell translation stops. Virus brings in its own proteases. These proteases will activate host cell proteases. These host cell proteases cleave this ribosome protein called EIF 4F that is 220 kilodalton. That's the molecular weight of it. Then, when this protein is being cleaved, there would be no cap binding complex for host cell messenger RNA, no attachment of host cell messenger RNA to host cell ribosomes. Therefore, there would be no host cell protein synthesis. And if there is no protein synthesis, which is the essential components of the host cell for its survival, this will lead to the death of the host cell. So poliovirus release is accomplished by host cell lysis, slide number 22. So the release of the poliovirus occurs within 5 to 10 hours in cell culture. Each virally infected host cell releases anywhere between 25 to 100,000 viral particles. This is when only one virus comes in. So the process is pretty effective. And not all viral particles that are being produced are infectious. Remember, only those that are positive sense. And virus spreads to other cells, and about 1% of these infections will go into the central nervous system. So, what will happen during poliovirus infection? The entry is through aerosol or ingestion. Virus replicates in the oropharyngeal tonsils. Those tonsils are lymphoid organs that are located in the oropharynx region. Through that, we have primary viremia, where virus gets into the circulation. And after primary viremia, we have secondary viremia. That virus goes to target tissues, causes polio. It can go to meninges, cause meningitis, or in rare instances, it goes into the brain, central nervous system, and causes encephalitis that causes paralysis of the individuals. Franklin Roosevelt, the 32nd US president, and he himself was a polio survivor, so he was a strong advocate for a vaccine against poliovirus, and he said that the fight against infantile paralysis is a fight to the finish, and the terms are unconditional surrender. Another family is Calciviridae. The genus would be norovirus. These are viruses that have cup-shaped depressions on their capsid. They're very similar to picornavirus. Their genome is positive RNA, 7 to 8 kilobase, like picornavirus that was 7.7 in case of polio. And they have slightly larger capsid than picornaviruses. An notorious virus within this family, within the genus norovirus, is Norwalk virus. That Norwalk virus was discovered by electron microscopic examination of stool samples during an outbreak in Norwalk, Ohio, where they found that they are very small and round viruses. Norwalk virus is the major cause of epidemic viral gastroenteritis. The mode of transmission is through a fecal oral route from water, food, shellfish, eel food service workers can transmit the disease very efficiently. It usually occurs in restaurants, nursing homes, schools, daycare, daycare centers, vacation settings, including cruise ships, and in uh, United States, 50% of the epidemic viral gastroenteritis per year is due to Norwalk virus, so about 23 million people are infected annually. Another family of positive strand RNA viruses are is the family of Flaviviridae. A prime example would be yellow fever virus, again positive RNA, about 10 kilobase genome, slightly larger than the previous two classes we discussed. It has, again, like polio, one single open reading frame. Unlike poliovirus, at the 5' end of the genome, it has a 7-methyl guanine cap. At the 3' end, like polio, it has a 3' poly A tail. Just like polio, this single open reading frame will be translated into one single polyprotein that will be further cleaved through proteolytic cleavage to make structural, such as capsid proteins, M protein, E protein, and non structural proteins, which are a group of NS for non structural, one, two, three, four, five. And some of these, like non structural 2 or 4, will be further cleaved to become NS2A, NS2B, NS4A, NS4B. Slide number 26 is a little bit more description of the yellow fever virus, which is part of the family Flavivirus. Slide number 27, another genus within the family Flaviviridae, is the genus Flavivirus. In the past, they used to call it group B arboviruses. There are a number of viruses within the family of flavivirus. These include yellow fever virus, uses monkeys as host. Dengue virus, it causes a fever that is so painful they call it break bone fever. As a matter of fact, we have a case study on dengue virus. It is an important virus because about 50 million cases are found annually worldwide. Another virus within this family is called West Nile virus, uses birds primarily as its host, and hepatitis C virus. Remember at the beginning, I said that these flaviviruses were formerly called group B arboviruses. So these are arboviruses that they need an arthropod for during the process of their life cycle. However, hepatitis C is not an arbovirus. Its transmission is done by blood and body secretions. Another family of viruses within the R positive RNA group is called Toga viridae. Toga means coat. They are positive RNA viruses. These are enveloped viruses. Icosahedral capsid, a little bit larger than picorna, and they replicate in arthropods, various animals, and various and humans. So it has a broad host range. A genus within the family of Togaviridae is called alpha virus. formerly called Group A arboviruses. A prime example would be Synbis virus that uses birds, monkeys as hosts, and rarely it causes mild diseases in humans as well. Another virus called equine uh, encephalitis virus that uses horse, birds, rodents as its host. And we have three different types of this equine encephalitis. We have Eastern, EEE, and Western, WEEE, and VEE, or Venezuelan. Okay, the first one, EEE, causes diseases in eastern United States, in the Atlantic region, and in southern United States. There is 30 to 70% mortality rate. WEE causes disease in the western United States or the Pacific region, Southwest also. 3 to 7% mortality rate. As you can see, it's like 1/10 of or 90% less than Eastern queen encephalitis virus. And the Venezuelan, we see it in Venezuela, South and Central America, and it has rare fatality rate, so it usually doesn't kill. So it's the least severe of the three types. Another genus within Togaviridae family is ruby virus, ruby red. A virus in this disease, in this group, is called the rubella virus. Rubella virus causes 3-day measles. Transmission of the rubella virus, the causative agent of 3-day measles, is by direct contact via aerosols. However, no arthropod vector plays a role in the transmission of the virus. Rubella virus, or the disease measles, is characterized by mild skin rashes. However, pregnant mothers can transmit the disease to their fetus, and upon transmission of the fetus, it can either cause congenital defects or it can cause the death of the fetus. Slide number 35 talks about the genome structure of Synbis virus, which is a toga virus, and the genome is positive sense RNA. The genome is 11 kilobase positive RNA. It has two open reading frames. As you can see, one open reading frame one is about 7 kb. The second one is about 4 kb. 7 kb usually transcribes for replicase genes, and ORF2 usually transcribes structural genes. So again, the genome is a positive RNA, 11 kb, translates open reading frame one for polyproteins, multiple proteins. However, if the polymerase, if the replicase cannot recognize this sequence called suppressible stop codon, if it recognizes suppressible stop codon, so we only have a portion of ORF being transcribed for a polyprotein, but if the suppressible stop codon within open reading frame one is not being recognized, it makes a larger protein, the entire region, as well as it makes the polymerase. It also synthesizes a subgenomic messenger RNA, small fragment of RNA that is about 4 kilobase, and it can translate open reading frame 2. Slide number 32 is a little bit more description of Symbis virus genome. Slide number 33 talks about Symbis virus early gene expression and the production of a genomic RNA called 49S genomic RNA. So again, virus attaches to specific receptor. Through receptor-mediated endocytosis, it gets into the host cell cytoplasm, uncoats separation of genome and capsid, and release of positive strand RNA genome of Symbis virus into the cytoplasm. Since it's positive, it can bind to the host cell ribosome and translates open reading frame 1 for a polyprotein. And this polyprotein is being cleaved into 4 non-structural proteins. Non-structural protein 1, non-structural protein 2, non-structural protein 3, and non-structural protein 4. The issues that we just discussed are depicted in slide number 34. Slide number 35. Let's talk about Symbis virus transcription of complementary RNA strand. Remember, we said that the genome has 2 open reading frames, 5' cap, 3' poly A tail. It's a positive genome, so the positive genome will serve as a template and will be copied into an anti-genome RNA, which is a negative strand, and this process uses early viral proteins and enzymes from open reading frame 1. So those enzymes called replicases that are encoded by open reading frame 1, this region, will come and start making a negative strand off of the positive template. Slide number 36, Symbis virus replication genome RNA, or RI2. The 3' end of the anti-genome that we made during the first phase, which is the 49S negative RNA strand, is used to make more genomic RNA, which is the positive sense. And anti-genome uses viral replication enzymes to make positive RNA genome, and the positive RNA genome, the newly synthesized positive RNA genome, again serves 2 purposes: messenger RNA for translation of into the proteins, as we discussed in the previous couple slides, as well as it can be used as the genome for new virions or viral particles. This is the early gene expression, then replication of the genome. Now we talk about viral Symbis virus late gene expression, which leads to the production of a 26S subgenomic messenger RNA. Subgenomic is just basically a smaller segment of the original messenger RNA. So what will happen? Virus brings in an enzyme called viral RNA replicase. It's a virus, a replicase that can replicate viral RNA. This enzyme binds to a region on the antigenome RNA, the native strand that we made. This region is called interior replicase start. So viral RNA replicase binds to the interior replicase start site. It's a region on the negative sense RNA that we had made, or the antigenome that we made. This will result in the production or synthesis of a subgenomic mRNA, or 26S, that this will code for open reading frame 2. An open reading frame 2 will be translated to make structural proteins required for capsid and envelope. And this messenger RNA, the subgenomic mRNA that is being produced, has 5' cap and 3' poly A tail. Slide number 38 is just a depiction of what we just discussed. Slide number 39, SYNBIS virus RNA. There is a temporal regulation of viral RNA synthesis. Temporal meaning time-wise. So within the first 3 hours upon viral infection, we will make both positive and negative sense RNA strands. And after 3 hours, we start producing mainly positive strand RNA, and we make 10 times more of subgenomic mRNA 26S that codes for open reading frame 2 than the genomic mRNA, the 49S. Slide number 40, the assembly and release of SYNBIS virus. SYNBIS virus is being released from the host cell by budding. Initially, those glycoproteins, viral glycoproteins that will be incorporated into the viral envelope, are glycosylated in the host cell endoplasmic reticulum and Golgi apparatus. And after the glycosylation, so these glycoproteins will migrate to a specific region of the host cell plasma membrane, or PM. At the same time, due to the translation of open reading frames, capsid proteins are being made. Capsid is being assembled in the cytoplasm, and it incorporates the viral positive sense, or the 49S subgenomic mRNA in it. The new capsid that is wrapped around the virus will bud out of the cell through that region of the host cell plasma membrane that has been modified by the insertion of viral glycoproteins. During this process of budding, virus will pick up an outer envelope, so there would be a host cell plasma membrane wrapped around the viral capsid. Slide number 41 is just basically the same description of what we just discussed. Slide number 42, the family Picornaviridae, including I'm sorry, the family of Coronaviridae. Corona means crown, and it's usually named because of the structures on the surface of the virus that gives it the shape of a crown. coronavirus. These viruses are enveloped viruses. They have very large petal spikes, and they have a flexible helical nucleocapsid. Coronaviridae family of viruses are positive RNA genome, large 30 kilobase. It has a 5 prime cap, 7 methyl guanine cap, and at the 3 prime end of the positive RNA, we have poly A tail. This codes for 5 open reading frames as depicted by these rectangles. And as you can see, some of them are overlapping. And these open reading frames result in the synthesis of a number of subgenomic mRNAs. Slide number 43 is just a depiction of what we just discussed. Slide number 44, a genus within the family of Coronaviridae is coronavirus. Coronavirus causes common infection of human. The infections look like common cold. They have symptoms similar to common cold, gastroenteritis or GI infections, and lower respiratory tract infections of the lungs because of the pandemics and the widespread of coronavirus infection. There is a module dedicated to coronavirus, which includes selective video clips of various stages of coronavirus infection and the role of various treatments, including the vaccines, as well as there is a recently published scientific review article written by undergraduate students at Cal State LA Biological Sciences. And we also have in this genus, we have SARS-CoV. SARS stands for severe acute respiratory syndrome. It causes fatal pneumonia. We have MERS also. MERS-CoV. MERS stands for Middle East Respiratory Syndrome. This also causes acute respiratory infections. Is a very highly fatal infection. We also see these infections in mammals, other mammals, as well as birds. So, lecture summary: RNA virus genome replication requires a double-stranded replicative intermediate. Since what we discussed so far are positive strand RNAs, they need to make a negative strand RNA, and that negative strand can serve as template during the second phase to make an RNA, a positive RNA. So it goes through 2 RI or replicative intermediate phases. Poliovirus RNA genome has only 1 open reading frame that codes for 1 polyprotein, and it will be further autocatalytically cleaved into 3, and then further proteolytic cleavage into many proteins. Other positive RNA viruses include norovirus, yellow fever virus, dengue virus, West Nile virus, hepatitis C virus, Sinbis virus, equine encephalitis, rubella virus, and coronavirus. Sinbis virus has 2 open reading frames that are expressed using two messenger RNAs. These two messenger RNAs, one is 49S genomic, and the second one is 26S subgenomic messenger RNA. The reading materials for this set of lecture slides come from chapter 14, Replication of Positive Sense RNA Viruses. And there are a couple other slides that I want to discuss about complete blood cell count, or CBC. Viral infections usually cause an increase in the number of white blood cells, in particular lymphocytes. So a CBC, or complete blood count, determines the number of red blood cells, the number of white blood cells, and differential count, which basically tells us which of these 5 different classes of uh, uh, white blood cells are increased or decreased. It gives us an absolute number of the different types of white blood cells. That from the top, we have red blood cell, then we have a granulocyte, which in the cytoplasm they have granules, including neutrophil. Second one is basophil, that produces histamine. Neutrophil participates in phagocytosis. The third one is eosinophil. That eosinophil is increased during allergic reactions and some parasitic infections. Then we have agranulocyte white blood cells, that their cytoplasm do not contain uh, any granules. These include monocytes that participates in phagocytosis, and smaller lymphocytes that have a large nucleus. The nucleus of monocytes looks like a kidney bean, whereas the nucleus of lymphocyte looks like a sphere. We have 3 different types of lymphocytes: B cells, T cells, NK cells. B cells participate in the production of immunoglobulins, or antibodies, humoral immune response, whereas T cells, we have CD4 and CD8 primarily, that participate in cellular cell-mediated immune response, or cellular immunity. And lastly, we have platelets that participate in blood clotting. So, increase in the number of leukocytes is referred to as leukocytosis. We usually see leukocytosis in bacterial infections, where PMNs, polymorphonuclear cells, or neutrophils will be increased, the first type of white blood cells. And some diseases, and even some viral diseases, cause a decrease in the number of leukocytes. We call this leukopenia. And differential white blood cell count is basically visualizing these under the microscope, is when we count 100 white blood cells only, and then obtain the percentage of each type of white blood cell. Slide number 50 is a table of normal values of cerebrospinal fluid, or CSF, since some viral infections and bacterial infections can get into the CSF and cause infections there. Therefore, it is important to know what's going on with CSF very briefly. So when we have an infection, infections can usually cause the migration of leukocytes into the central cerebrospinal fluid, followed by an increase in their number in the CSF. And therefore, it is very useful to differentiate between viral infection and bacterial infection. One way I said that usually during viral infections, we have more of lymphocytes. In bacterial infections, we usually have more PMNs or neutrophils. Another way of distinguishing between viral and bacterial infections is via conducting a biochemical analysis, measuring the glucose as well as protein contents of cerebrospinal fluid or CSF. If there is no change in the glucose, it, yet a decrease in the glucose content of CSF, it suggests uh, no change would be viral infection because viruses do, unlike bacteria, they do not consume glucose. They use host cell materials and nutrients. So no change in CSF glucose, possibly viral infection, decrease, possibly bacterial infection. If there is no change in the protein content of CSF, suggests uh, viral infection. And if there is an increase in the protein content of CSF, it suggests bacterial infection due to the growth and due to the growth of bacteria. Therefore, we can assume that if there is no change in the glucose or protein content of CSF, it suggests viral infection, but it needs to be confirmed by culturing CSF, looking at it under the electron microscopy, as well as looking at the type of white blood cells inside of the CSF. This is the end of lecture 6, positive RNA viruses. Thank you very much for your attention.